And what I want to particularly focus on are the methodologies associated with DNA methylation. So, when we think about genetic analysis of a crop, we can think about the changes in genotype that we evaluate across different lines. But when we think about the way that a plant responds to its environment, we think about the way a plant alters its gene expression without altering its DNA sequence. And so, to select for crops that have the capacity to respond to their environment, we need to use epigenetic data sets rather than genetic data sets. So, epigenetics is the heritable alteration of DNA that influences gene expression but not DNA sequence. And epigenetic marks are generally associated with histone modifications, where there are particular limitations because to look at histone changes in a crop, we have to rely on antibody detection, and that often leads to bias. And it's a less comprehensive assay. You can only see a small subset of changes that occur in any given assay. But if we work with DNA methylation as an epigenetic mark, we have the capability of looking at single nucleotide resolution. Every single cytosine within a genome can be assayed. So there is a certain advantage to working with DNA methylation because it is a simply derived data set, as simple as sequencing, and it is a very complete data set, as complete as the complete genome would allow. But there are limitations to DNA methylation, mainly associated with current analysis procedures. So methylation data is noisy data. It means that because we derive DNA methylation information from multiple cell types at a single time, there will be heterogeneity at any given site. And we need to be able to work with that heterogeneity and to interpret from it. What we also need then are more sensitive data analysis methods that allow us to resolve meaningful biological information that tells us what genes are involved in a crop's response to its environment, deriving from methylation data without being hindered by the noise in the data set. So DNA methylation, as well as histone modifications and non-histone proteins, define chromatin structure, and ultimately they regulate gene expression. So if we can identify those regions of the genome that undergo change in DNA methylation pattern, we can use that information to identify the regions of the genome that are responding to environment and providing phenotype. So when we think about methylation data analysis, one of the challenges that we see is that methylation is heterogeneous in our data sets. So what that means is that if we were to read several, if we were to derive a data set, it will be full genome and it will be deep, which means for any given cytosine, you'll have maybe 20 to 30 data points. The problem is that you might have as your control a cytosine with 30% methylation, and in your treatment, that same cytosine is 70% methylation. And your challenge is to decide, is that a change or is that no change associated with my treatment? This type of complexity has led people to develop methods that use arbitrary cutoffs for any given site to decide whether you call this methylated or whether you call it non-methylated. Often, using a bisulfite sequence data set, you'll use a Fisher's exact test with a p-value adjustment problem that will make it will make more difficult your ability to identify those regions of the genome that have small levels of change. So what do I mean? I mean that if you're going to read a data set for methylation, you need to be prepared to understand that some changes will be large and some changes will be very small. And they're both important to your analysis. If you use the standard conventional methods for analysis, they are designed to only detect the very large changes. Large changes at individual cytosines and large numbers of changes within a given region. And so what that does is to immediately remove from your data set the very subtle changes in methylation that you might care about. So one of the conventional methods for methylome analysis is to look at differentially methylated regions. This means that you use an arbitrary region of the genome in order to identify an arbitrary number of changes, and then you can arbitrarily decide if all those changes are in one direction to methylated or in another direction to non-methylated. All of those arbitrary changes or arbitrary decisions by the user means that you have a much less robust data set and much lower resolution for looking at methylation changes that are subtle, that are not large in magnitude. And those subtle changes are generally the changes associated with gene expression. So there are equations that one can use for calculating overall methylation at any given cytosine and then as a data set. The original methods for analyzing DNA methylation usually focused on methylation mutants. These are all different mutants within genes that control methylation. And you're looking at a heat map that shows you that there's large numbers of significant methylation changes that are associated with these mutations. But if you develop your method by using these types of mutant lines, your method is automatically biased for looking for very large changes. And often your methodology is not suitable or sufficiently sensitive for identifying changes within genes that are going to be very low density. And those are often the changes that are associated with environmental response. So when you want to study changes in the epigenome in response to drought or response to high light or response to cold, you often will not see any of those changes if you use these original conventional methodologies. So the method that we use is designed specifically to look at environmentally induced epigenetic change. And the way that we think about methylation is not on an individual cytosine with an arbitrary cutoff approach. What we do is we think about methylation as a probability distribution. And because it forms a distribution, you can look at methylation under the curve associated with background noise. And you can also derive a separate distribution for methylation that's associated with your treatment, drought, cold, a genetic mutation, whatever you're studying. And understanding the relationship between these two distributions of background noise, which you can learn by comparing controls, And treatment, which you learn by comparing treatment with controls, it gives you the opportunity to identify those methylation changes most likely associated with your treatment of cold, heat, or gene mutation. So I'm showing you in the lower right-hand corner how we imagine this to look. You have a distribution for control. That's all the methylation that occurs when you compare one wild type with another wild type with another wild type. And you take all of the variation in methylation in those controls and you subtract that from the methylation that you identify from treatment. So that would be your drought line with your drought line with your drought line. And what that gives you are two distributions largely overlapping. And where they do not overlap, out at the end of those curves, are where the differentially methylated positions will reside with highest probability to be associated with your treatment. So in this way, we can look at only those methylation changes that are clearly associated with what we're interested in, drought or heat, and we can remove all the background noise that we can also find within controls. And then what we can do is we can do this analysis with about 60% of our data to derive treatment-associated methylation changes, and then we validate with the remaining 40% by machine learning so that we know that we have high probability of identifying those methylation changes that only occur in our treatment sample but do not arise in our background control. We call this the Methylated platform, and it's available, publicly available, and that platform allows us to now analyze DNA methylation with much more sensitivity. So what we have to do to test that methylation platform is to now work with a system that gives us a very clear, unambiguous, and reproducible way to create epigenetic variation. And this is not easy because epigenetic variation created by environment, drought or heat, is very difficult to reproduce with fidelity. So what we use is a genetic mutation for our system. So I will show you in Arabidopsis, but what I'm telling you can occur in any crop species, not only Arabidopsis. If you start with a wild type plant and then you mutate or you suppress the expression of a gene called MSH1, is a perturbation or a destabilizing of the chloroplast genome. And that instability of the chloroplast genome behaves to the plant like environmental stress. So now we're creating an artificial way to create environmental stress. And the MSH1 mutant will respond by slower growth, delayed flowering, and sustained stress response. All of these plants behave as if they are subjected to heat and cold and high light and drought and biotic stresses. In fact, some of them will even transition to a perennial type growth habit. So many, many gene expression pathways are impacted by this one mutation. The reason we know that this is an epigenetic effect is that we have developed a protocol for validating epigenetics. And that protocol involves using a transgene, like an RNAi, to suppress MSH1 so that in the next generation, we can segregate away the transgene and ask if the effect is still present. If the effect is present, then it is an epigenetic change. And this we refer to as stress memory. So whereas the MSH1 original mutant is epigenetic state 1, the derived memory from that mutant we call epigenetic state 2. Now we can take an MSH1 mutant that causes epigenetic change, and now we can use a second epigenetic assay, and that's grafting. So now we can take MSH1 as the rootstock, wild type as the scion, and create a graft and derive seed from that graft. And those seed will give enhanced vigor and enhanced growth and enhanced yield and resilience relative to wild type. So now that creates a new epigenetic state that we call epigenetic state 3. We can also create an epigenetic state by crossing MSH1 to wild type, and that also gives us enhanced vigor. So now we have the ability to reproducibly create epigenetic state 1, epigenetic state 2, epigenetic state 3 through grafting, and 4 through crossing. And all of those will give us differences in DNA methylation output. And so we can compare all of those states relative to wild type to ask, how did methylation change for any given state compared to wild type? We validate this system by testing it in more than one species. So we can recreate this in tomato, soybean, sorghum, all of these different crops. But we also validate this system by demonstrating that it is dependent on the RNA-directed DNA methylation pathway. So we know that for the induction of the MSH1 state 1, you must have histone deacetylase 6 present. This is a component of the Rddm pathway, of epigenetic pathway in plants. We know that the methyltransferase DRM2 must be present for memory. We know that dicer-like 2, 3, and 4 must be present for the graft progeny changes, which say that what's being transmitted in the graft is small RNAs. And those small RNAs are no longer transmitted if the Rddm pathway is not functioning. So we know that all of these changes we've created are epigenetic changes, not genetic changes. And we know they're all dependent on the machinery of the Rddm pathway. The reason that this is valuable for our study of methylation is because we can now take rootstocks that are MSH1 and rootstocks that are DCL234 and MSH1 and compare the outcomes. So when we use a rootstock with MSH1 rootstock, we will see changes and enhanced vigor and change alone. If we use an MSH1 DCL234 rootstock that can no longer produce small RNAs, then we no longer see in the graft enhanced growth, and we no longer see those methylation changes. So a comparison of those two data sets allows us to identify unambiguously the Rddm targets within the genome, those genes that are undergoing targeted methylation change. So this system is extremely powerful for the validation of a methylation analysis platform. So the Rddm pathway is a very complex pathway that is responsible for production of small RNA signals and is responsible for targeted methylation changes. And so as I pointed out in this pathway, the MSH1 system we know depends on HDA6 here, depends on DCL3 here, and depends on DRM2. No doubt it depends on other components, but those three mutants are especially valuable for demonstrating for us that the changes we're looking at are dependent on de novo targeted DNA methylation. So what is the system that we can use now to understand how to analyze methylation data in plants? Well, first you start with the MSH1 system. It gives us 4 epigenetic states, and it incorporates Rddm mutants for functional verification. And then we combine that with Methylated, which is the system I described that evaluates DNA methylation as a probability distribution. It measures and subtracts background variation. And so by using signal detection, it allows us to enhance signal to noise ratio that allows us to be very sensitive in finding changes in methylation associated with genes and gene networks. And then we incorporate machine learning to assess our accuracy for differentially methylated positions. And then there is protein-protein interaction network analysis. Once we find the genes that are differentially methylated, we can put them into networks to identify what functions those networks control and what phenotypes we expect from the plant. So this takes us from the individual perturbation to the genome-wide analysis to the identification of networks associated with phenotype. Now, if we were to just look at gene expression, we know that phenotypes are changing because in the left panel in each of these pathways, we are looking at what happens with enhanced growth from grafting progeny. On the right, we're looking at the original MSH1 mutant. And what you see is by coloring, red means higher gene expression, blue means lower gene expression. You can see that for each of these pathways, we see an antagonistic relationship of growth versus stress response. So we know that the plant is undergoing a change from growth to enhanced sustained stress response in these materials. The problem is that we don't have a way to understand the control mechanisms by just doing gene expression. It's not enough information. Now, if we look at differentially methylated genes, those are the genes that we derive that undergo treatment-associated methylation changes for state 1, state 2, state 3, and state 4. We can overlap those data sets and say which genes are in common for each of those states, okay? And there are 871 genes that all undergo methylation repatterning in response to the MSH1 perturbation. Now, the reason we do that is because if you can identify genes that undergo methylation change, you can now ask whether different methylation changes, if they're going to give you growth or if they're going to give you stress response. And the way we do that is by hierarchical clustering of those data sets from those 871 differentially methylated genes. I'm now able to look at total variance for those differentially methylated positions that we identify for each of those data sets. And what I want to show you is that all of the methylation changes for those 871 genes for state 1 are more closely related to one another than they are to the methylation changes within the same genes in epilines, state 4, or memory, state 2, or graft, state 3. What that means is for those 871 differentially methylated genes, the changes that occur are state specific. It means that each state that we see, each phenotype we see, has a different methylation pattern for those same genes. They all undergo change, but the change is different depending on the state we create. This is important evidence to say targeted methylation changes can define phenotype. If we were to take those 871 genes and now integrate them into gene networks, what we find is that they fit within, that 531 of them form one very large gene network. And of those 531 genes, if we were to compare our data sets for MSH1 and for MSH1 DCL234, where it's no longer producing small RNAs, you will find that everything in orange is targeted by the Rddm pathway. And that comprises 77% of these genes that we know to be targeted differentially methylated genes in response to MSH1. So that means that those only occur when small RNAs are present, and those small RNAs find a cluster near this gene that causes that differential methylation to occur. So now we're able to say a very large proportion are targeted, and they're targeted differently if we're looking at memory or stress state, and if we're looking at enhanced growth. Now we can use a k-means cluster analysis, which gives us sort of an unsupervised machine learning approach to identify those genes within those 871 that are most likely to be hubs within those networks based on their connectivity. And so if we look at we identify 67 genes out of the 871 that are hubs, we now find that we can identify the gene networks that are the most important to defining phenotype. And those gene networks are response to stress, which we expect because MSH1 and memory look like they are sustained in stress. We also find developmental process associated with growth because we're looking at vigor. We also will find gene expression, spliceosome, histone modifications, and chromosome organization. These are all networks associated with epigenetic reprogramming. These are chromatin modifying components of the Rddm pathway that regulate gene expression based on methylation. So what's important then is we can now start to identify individual genes that are defining phenotype, in this case, in response to an artificial stress environment. But the same thing would apply if we were doing this in response to drought or response to heat or cold. So for example, we find splade. Splade is a central component of the decision that a plant makes to become vegetative or reproductive. So the timing of flower. And splade works with a very important component right next to it called Brahma. And they work together to define for a plant its growth vigor as well as its decision for flowering. TOR is a gene also central to this component. It is the target of rapamycin, which is a regulator of cell growth in response to nutrient availability. TOR is the decision point for a plant to enhance stress response or enhance growth vigor. So these genes are not arbitrary, they're central to defining a plant's decision for vigor or for stress response. And there are many others in this group. Other major ones are the component of RNA polymerase II, the UBP26, the NRPD1. These are components of the Rddm pathway. All of these are major central hubs of these gene networks that are controlling the process of growth, but they're doing so in association with differential methylation that is state specific. More than 81% of these components, 81% of these components are known to be direct de novo Rddm targets. You can see that because they're shown in orange. All of these undergo differential methylation in response to MSH1, and that methylation pattern will differ depending on state, growth state or stress state. Now, when you do gene network analysis, one of the problems that will arise is that networks are biased by the way that they're developed. The gene networking information we have for plants is based on multiple data sets, some of them co-expression, some of them protein-protein interaction by yeast-2 hybrid, some of them through the literature parsing to say that two things are found together or seem to be operating together or show some sort of epistasis. And so the difficulty is that creates a certain amount of bias through the literature. So one way to validate the gene network data that we derive from methylome is that we use weighted correlation network analysis. And by using weighted correlation networks, it allows us to develop the networks without annotation, so that you're doing this blind, using other weighted components to tell us these two factors are found together and likely to coexist or to co-function. And using weighted correlation network analysis, we find more than half of those 67 core hubs are present here. They're shown in red. So this actually helps us to validate that the networks that we're identifying are likely to be correct. Once we can identify DNA methylation changes that occur within genes that are associated with phenotype, we can now start to look at where those are occurring within the gene. And the way we do that is by identifying sites that we know are targeted sites of methylation and then looking for evidence of motifs around that site. And here we're just testing one of these motifs. And what you're looking at are four sites within the UBP26 gene. And what you're looking at is state 1, state 2, state 3, and state 4 data sets. And what you find is that for this particular site within the gene, it is only methylated, here in orange, in memory. But at this particular site within the gene, it will be methylated in the MSH1 state 1 or in the graft outcomes. But if we look at this particular set of sites, they will only be methylated in state 4 from crossing. So we're seeing state-specific changes in methylation that we can identify. We can also show that those methylation sites disappear when we add DCL234. So it's methylated, and then when we add DCL234, that methylation disappears. So we can discriminate those sites that are dependent on small RNAs. And as you can see, we can identify several of these clusters as methylation sites. These are target sites within genes that are responsive to environmental change. Now, I kind of want to finish with an even more interesting aspect of the way methylation works. What I've told you up till now is our ability to read methylation patterns within genes that allows us to identify those genes as being important to phenotype. It's really valuable, but it only focuses on a small proportion of the methylome data. So as I said earlier in that little slide I showed you, we think of methylation as a probability distribution, and we're only reading the methylation out here on the tail end of this curve as being treatment associated, that which is caused by MSH1 or that which is caused by drought, cold, whatever you might be testing. But there is a very large, major portion of the methylome that actually resides and overlaps with control. And within that is also important information. This background noise, if you will, actually has meaningful information. And what we use is we use what's called maximum entropy modeling in order to understand this. And I don't want to go through the mathematics of this, but understanding maximum entropy, or let's say information available, and combining that with a calculation of molecular machine channel capacity, which is only estimating the input of methyltransferases and demethylases as enzymes or machines that are adding or subtracting energy in the system, gives us the ability to derive a mathematical equation for system entropy, which has both classical entropy as well as what we call molecular machine contribution, the enzymes associated with methylation and demethylation during Rddm targeted methylation change. And by doing this, what it gives you is a printout, essentially, an ability to estimate loss of information as well as free energy of the system, and to look at the magnitude of loss of information or the magnitude of the loss of free energy when you make a change in the methylome. And let me give you an example. So here you're looking at 5 wild type samples, and you're looking at the entropy associated with chromosome 1, 2, 3, 4, and 5 methylomes. So we're only looking at methylation at one chromosome at a time. And what I want to point out is, for example, if I compare wild type and then scroll down here to memory, this is memory generation 3, you can see that there is a change and a loss of information associated with the transition from wild type to going to memory. More importantly, we can use non-memory, which is when we have silenced MSH1 and then removed our transgene, but we still see no phenotypic change. And what I want to point out is with non-memory, we see 2 standout lines. All of the others are approximately the same entropy level, and then there are 2 lines that are clearly distinct. When we take non-memory plants and we grow their progeny, only those 2 lines, number 3 and number 5, transition to a full memory state. So these entropy values were predictive of those plants that were not yet stable and were in transition toward memory. So we know now that we can use this information for prediction. What I also want to point out is then if we come down here to comparing, for instance, memory generation 1 and memory generation 3, what you find is that things become more stable by the time you reach generation 3. This is memory giving rise to memory, giving rise to memory. 3 generations, it's the same lineage, but as you can see, it is losing more and more information and free energy in this new state. What I also want to point out is if you look at this wild type and you compare it to the wild type on the top, they're both Columbia 0. The difference is significant in the entropy values. And the reason for the significant difference is that this wild type on the top is grown 12-hour day length, grown in pots, grown in a growth chamber, and grown in normal media. These wild types down below are grown 24-hour day length. They're grown in on agar with a nutrient media, and they're grown only to small seedlings. So their growth conditions actually create a distinct state in their methylome as a consequence. And what I also want to show you is down at the bottom. Here we're using a MET1 mutant. MET1 is a gene, it's a methyltransferase that is responsible for CG methylation throughout the genome. It's a very significant change to methylation. And as you can see, the changes in free energy and the changes in information loss are much, much more substantial. So we can actually derive quantitative information about the state of a system and how disorganized or dysfunctional that state is, or how stressed the state is by virtue of total methylome data using this methodology. So I just want to summarize what I've said here. This is looking at intragenic methylation. It's environmentally responsive and it's low density, meaning there are not large changes, but small changes. We can look at environmentally induced epigenetic changes and how they impact gene networks using these methods. We can then take advantage of RNA-directed DNA methylation mutant analysis, and that allows us to confirm the changes that we see to be real. And then we can use maximum entropy-based analysis that allows us to predict the outcomes in phenotype for the kinds of changes we're looking at. So how would one use this kind of technology? We would use this technology as we're trying to understand, as we breed crops for more and more resilience to changing environment. We want to breed crops that have the capacity for resilience. That can be learned through methylome analysis. We want to learn those plants that go into a very different state under stress than under normal conditions. That we can learn from methylome analysis. And we want to learn how to identify those gene networks that are most important to plant resilience so that we can breed for enhancement of those network functions. That we can learn from DNA methylation analysis. So we can use this system in the field. This is looking at MSH1 graft progeny. They're enhanced for growth, enhanced for resilience. They give somewhere between a 10 and 20% increase in seed yield for canola using this system, strictly epigenetic, not changing genotype at all. And with that, I just want to thank the people who contributed the work that I described to you today. Isaac Dopp is a graduate student who studies the HDA6 interaction with MSH1. Xiaodong Yang is a former postdoc in the lab. He now has his own laboratory in China, but he collaborates with us. He did the memory analysis. Dr. Hardik Kundaria does all of the epiline and the graph studies. And Dr. Robercy Sanchez is the person who developed the Methylated platform that I described. We work with a small startup company called Epicrop Technologies, and they are responsible for moving this system into crops and testing it in the field. And we also collaborate with Mike Axtell, who works with us on understanding small RNA changes in plants and how they impact phenotype. And these are my sources of funding for this work. So with that, I want to thank you for the opportunity to present this work, and I hope I'll have an opportunity to answer your questions.